Hello, everyone. I'm Sarah Jennings, and welcome to our SETI Live today. I'm here with one of our Frontier Development Lab teams. Frontier Development Lab is an applied AI research accelerator, and we bring together PhDs and postdocs from machine learning and pair them with space scientists. And we basically go on an 8-week journey working on problems in Earth science and space and exploration. So today I have with me the astronaut health team. We're about at the conclusion of week 5, so we have a few more weeks left. So I would like to welcome our astronaut health team, Linus, Giuseppe, Paul, and Aaron. Thank you guys for taking time to talk with us today. So to get us started, I would really like to hear a little bit more about the challenge that you're working on and why that challenge is important.
Sure, I can introduce that. So, hi everyone, I'm Aaron. I'm an MD PhD student, but I love working in AI as well as that little bit of medical side of things. So I will start by, showing a few slides to help people understand what we're up to. So we have been on the topic of the space medic for causal inference for added distribution generalization. Now, there's a lot of long words in that. I didn't like the title looking at it first time too, because it scared me to death. But I will unpack that to show you a bit of what that actually means. So this is the team. There's me here, of course, and also joining us in this call are Giuseppe, Linus, and Paul. We've had amazing faculty supporting us from NASA, Intel, and the Mayo Clinic, as well as Lancelot's team. So let's talk a bit about space and the biological challenge of it. Going to space is really hard, not just getting off the actual ground, but being in space is in itself a very inhospitable environment. And one thing we are really interested in long term is radiation exposure. So just by being in space, you are bombarded by heavy ion particles, which are radioactive, and this kills cells. It causes cancer, it causes mutations, and this is not great. And it presents a real risk to humanity's survival as in long-term spaceflight, because if we, fly up to space and in 3 years we've got enough radiation that we can't come back down again, that's going to really put a hamper on how much space travel we can do. But interestingly, this problem is also quite relevant to the realities of what we see more in day-to-day life right now. So for people who work in aviation, they're exposed to a lot of radiation for the same reasons. By being higher up in the atmosphere, there's a lot more in the way of radioactive particles around and radiation states. And so we wanted to try and understand what might be driving diseases like cancer when you get irradiated. Because when you get irradiated, lots of mutations can happen, but some of them may not have much of an effect. Others may be really, really important. This is really interesting to us because it means that if we know what may be driving a cancer in response to certain radiation types, we might be able to design medications to stop it or design, better spaceships to help prevent this. And so this is a really important thing for us to think through. And so what we are interested in is the idea of causal inference. So can we say that like this thing X leads to you having cancer rather than some sort of random thing that happened that we have no idea that it caused that thing? When, as I said, if you get mutated, you may have mutations that always happen together with the cancer, but they may not be causing the cancer. And that's correlation not equaling causation. And so we're really interested in trying to understand this. And this leads us to thinking about how we do this in space, because the environment of space is very different from the environment of Earth. If I have patients who are living on Mars, they're in a different microgravity, they're in different radiation environments, they're going to look very different from the patients who live on Earth and different from the patients who live on the ISS. And so there's an open question of how can we try and combine the learnings across all these different environments. We in machine learning call this distributional shift, the idea that there are different distributions of people based on where they are in the world or what sort of people they are. And we need to try and find ways of addressing that and still being able to do the things we're interested in. So in this case, it's causal inference. So can we do causal inference in distributed settings to try and discover things that are relevant to health? And that's a summary of what we're trying to build. We're trying to build methods that allow us to causally discover things when people could be on Mars, could be on Earth, and could be on the ISS, and work across all those data sets together.
Great. Thank you so much for providing that introduction to the challenge that you're working on. And it looks like we have a great audience. So thank you for those of you that are tuning in from Florida, Kentucky, Poland, Romania. Welcome. And if you do have any questions for us at any point in time, just share those with us, and we might have a little bit of time at the end to take some audience Q&A. So you had just talked a little bit about the work that you're doing, and I know that you guys are building off of some of the work that was done last year, and they developed an algorithm called CRISP. And I was interested if you could share a little bit more about that and how you are building upon the work from last year.
Yeah, of course. We have some slides for this as well. Thank you very much, everyone. So I'm Paul Duckworth. So I'm a postdoc at the Oxford Robotics Institute at the University of Oxford. And I'm just going to try and describe briefly what last year's astronaut health team worked on as part of FDL 2020. So last year they were tasked with identifying causal drivers of colorectal cancer. And so the idea is here that you really want to learn causal drivers, causal features, and not so much interested in the actual predictive performance of a particular algorithm, but the actual causal features are more important to the task. So this is a kind of a toy domain here, but imagine if you wanted to build or you wanted to learn the causal drivers of identifying which animals are in these particular images. So what you might do is you might go and grab a load of photographs of cows and photographs of camels, and you're trying to build a predictive model of which pictures or which photographs contain cows and which photographs contain camels. The problem here is that every time you take a picture of a cow, it's got a green background, a grassy background, and a blue sky. And every time you take a picture of a camel, it's got a kind of sandy background and a desert environment. So what you really want to do here is learn the causal features of which animal is in each image, and not just the kind of spurious correlations between the background pixels and the particular images that you have access to. So if you trained a standard predictive model on the data set that I've shown on the top left, and you receive some test images at test time, like on the bottom right, the cow on the grassy field, you'd probably predict that correctly as a cow. The cow on the kind of desert background, you'd probably say that was a camel. And the cow, sorry, in the icy snow background, you just have no clue what that was. Like, your model wouldn't be learning the actual causal drivers of this task at all. It would just be kind of correlating the background with the labels that you've provided. Next slide, please. So last year's astronaut health team developed a platform called CRISP, and that was based on this idea of invariance. So there's a link between invariance and causality that we can use for generalization. So the idea is to identify features that are consistently predictive across different environments. So here you might take pictures of cows on the grassy environment and put those into your model. Take pictures of your sandy the cows on a desert background with a desert environment, and you'd much more likely identify the actual important pixels in this problem. So then when you're provided with a cow in a kind of a snowy like background, you're more likely to be able to predict that in that challenging out of distributional kind of test set. Next slide, please. So yeah, last year's astronaut health team developed this causal relationship inference search platform. And it was an ensemble of multiple models where the data came from multiple different environments, meaning that they can tease out the causal structure in the data sets. It was implemented on Google Cloud Platform, and it was fully Dockerized for kind of convenience and ease of use. Next slide, please. So digging down into a few of the details. So the ensemble of causal methods that were developed was the deconfounder method, a method called invariant risk minimization, and invariant causal prediction. So the CRISP platform would run each of these models on the data sets, and it would have a voting and a combination mechanism and provide out the most likely to be causal features to the user. Next slide, please. Thanks. So yeah, the idea of this year's astronaut health team was that the stakeholders were really interested in the previous version of the CRISP platform, and it was widely picked up throughout NASA and the Mayo Clinic, who were the partners of last year's astronaut health team. And through kind of experimentation, they kind of discovered that we could take this slightly further. So starting kind of in the bottom left here, one of our tasks has been to enable a wider range of tasks, so a wider range of endpoints in our data sets. So things like multi-class classification or regression targets. Going to top left image, so the Mayo Clinic in particular were interested in out of distributional generalization. I think we're going to speak more on each of these later. Intel were specifically interested in the federated learning aspect. So can we learn these models, these causal models, if our data is siloed across different physical locations? And this idea of can we use radiated mouse data to inform our models about human astronauts, since the data sets we have on human astronauts are super small. And we've got more mouse data than human data. And that's kind of a brief overview of what the astronaut health team did last year and what we plan to do this year.
Thanks. Paul, that was really great. And I know that you guys are not using those cow images, and you mentioned some of the data that you're working with. Can you talk a little bit more about the data that you are utilizing, where it comes from, and what it looks like? Giuseppe?
Yeah, thank you. So I'll answer on this question. And I think, so the main aim of our project is to finally work with data on humans and on mice and try to transfer the learning between those two data sets. But as you might imagine, the big problem is that there's not an underlying truth, so that a known underlying truth behind these data. So it's really difficult to validate the methods that we're considering. So we are basing our work on CRISP from last year, who did already some experiments to show that their work was kind of well structured on some results. And we're trying to validate these results even further. So we are considering more strict and more difficult synthetic data on which to validate our results. So to know if we are actually able to discover the underlying causal structure in a problem. And the first data set that we use, it's a data set which tries to mimic the problem that Paul was talking about before. So where you are basing all your prediction on some features, but these are just really correlated with your solution just because of the sample you have. So basically what usually happens is that to make the task difficult to optimize on, to solve, you get that the test data, so the ones on which you base your accuracy, are basically all, for example, the cows on the snow. So making this kind of example very difficult. And the interesting thing is that we can then tweak these data as much as we want, so that we get the different problems, that like the different characteristics that we will see in the biological data later on. And so, yeah, this first data set that we use is like a synthetic one, and it allows us to validate our methods. And we also use a different kind of data, and Oran is gonna introduce them more in detail.
Yes, so what, of course, while Giuseppe has talked about the fact that we do have difficulties with working with, like real data, because we have the problem that if we, discover something that we think is causal in real data, we actually have to go out and test it. If we say, oh, we think this gene is really important in, driving cancer, we have to go out into, human cells and in mice experiments and actually say, okay, we've got to do lots of experiments on this. And so this may be a really big investment of time and money, and it may turn out very frustrating if it turns out that's not actually a causal factor. So we want to make sure our models work as accurately as possible beforehand, which is why we're so keen on using these synthetic systems where we actually say we know that this variable is causal in the set we've created. But then it means that when we get to the real data scenario, we still want to be as strong as we can. So firstly, we improve our models based on what we've learned from the synthetic data. But then secondly, when we get to the real world, we want to combine as much data as we can. And so this is where we've done some quite interesting stuff with CRISP. We are now using CRISP on both mouse data and human data at the same time. So there are some tissues in mice that are very similar to tissues in humans. So like blood cells and sort of the other blood tissues that you see, your red blood cells, your white blood cells, they actually look quite similar between humans and mice. There are some tissues that are very, very different. Of course, mouse brains are very, very different from human brains, funnily enough. There are other tissues, however, that can be quite similar at certain points. So maybe like lung tissue can look sometimes relatively similar to mice when you're looking at it from the human perspective. And so what we are thinking about is how can we actually work across these domains? Because in the same way you can think of a camel in snow being a camel in a weird environment, you can think of a blood cell in a mouse being like a human cell, but in a different environment where the environment is the mouse. And so we are learning across different organisms, but we're treating those as different environments. And it means that we can try and pull out factors which are still deriving those across those different environments. What this means, hopefully, is that we can get a lot more data. Because in the grand scheme of things, there haven't been that many people that have gone to space. But we have sent quite a lot of mice to space in little experiments. And similarly, we've got lots of data on mice that have been exposed to sort of radiation that stimulates cosmic radiation on Earth. And of course, I don't really want to go around irradiating people with cosmic radiation in the grand scheme of things. And so it means that we can do a lot of really powerful learning by combining this mice data with this human data and finding the commonalities that you couldn't otherwise find. And so that's a real value add that we're trying to create in CRISP right now.
That's really good to hear. And thank you both for the explanation about the different data that you guys are using. So health topics and challenges are, they have an extra layer of complexity. And I was just wondering if you had any particularly unique things about the health data that you're using and things that you're taking into account as you're developing your solutions.
Yes, thank you, Sarah. I'm Linus. I'm a PhD student in computer science. And as you mentioned, there are a couple of specific challenges to biomedical data. And since we're working with data from humans and mice, many of these difficulties also apply to our data sets. The biggest of these issues, and I guess this is also clearly a problem for us and also more generally in biomedical data, is the lower sample size. Odin mentioned it before, it is very difficult to obtain the type of data that we are working with. We cannot just go around and radiate people or mice just at will to produce our data, so we're restricted to relatively small data sets that already exist, either from space missions or from experiments on Earth where the radiation is simulated. Given that many of the causal inference tools that we are trying to use are relatively data hungry, this is a problem for our approaches that we have to tackle. And one of our approaches to do this here is to combine the mouse data, from which we have relatively much, with the human data, which is even more scarce. Another difficulty with the biomedical data is that it is very high dimensional. That means that for each sample that we have, we can measure its properties in many different ways. So for example, in our specific case, we're using so-called microarray data, which essentially gives us counts of gene expressions. And for each subject in our study, we have tens of thousands of genes that are measured. So we have only a relatively small number of samples, but for each of those, we have lots of data, which increases the complexity of the problem and makes it more difficult to identify the actual causal drivers. And then the third big problem that has also been briefly mentioned before is that the ground truth is unknown. So this makes it really difficult to validate that the results of our predictions actually are the true ones. That's also the reason why we go to synthetic data for our initial experiments. I wonder if you could go to the next slide, please. We also have the challenge in biomedical data that the data can be very heterogeneous. So that means that we can measure, as Odin mentioned again, for example, from blood or from lung tissue. We can also use genetic data or clinical metadata, which are all very different types of data that require different pre-processing and that have to be handled differently in causal inference pipelines. On top of that, we're also trying to use data from different organisms. So not only from humans, but also from mice, which then again amplifies this data heterogeneity. Yes, I wonder if you could go to the next slide. The last consideration that we also have to take is a bit more practical. This relates to the fact that biomedical data is very often distributed between different locations. Think about biomedical labs, which might have facilities in different countries or hospitals that are scattered across the country. Often it is very difficult to access all of that data in a centralized way, which would be ideal for any type of causal inference that we're trying to do. And a potential solution to this problem is so-called federated learning. Here we're working with our partner from Intel to train our causal inference models on data that is scattered across different data silos, so that we don't have to centralize the data from different locations, but can still gain insight from data that is distributed across different places.
Thank you, Linus. That was a really good overview of some of the complexities that you guys are handling with health challenges and health data. So now I'm going to move into a time where I'm going to ask all of you guys some questions. And I would really like to know why you are personally interested in this particular FDL challenge. Giuseppe?
Yeah, so, well, I think there's, first of all, the sheer fascination for this environment. So yeah, working on space problems is naturally something that, well, many of us have dreamt to do since they're kids. But besides that, I think the problem on from the mathematical point of view, that it's kind of what I'm really interested in, because naturally it's one of these problems that it's easy to explain to someone, but not really formally. And I'm kind of really fascinated by the formalism that we have to introduce in the space, and yeah, by how impactful this could be. Yeah.
Thanks, Giuseppe. Linus, do you want to go?
Yeah, sure. So I guess what I share with all the other guys is, of course, the interest in space that makes this problem interesting. In my day work, I'm trying to apply machine learning techniques to Earth observation problems. So this is also very space related. But on top of that, I also have a background in bioinformatics. So this is sort of a combination of both of my interests from the biomedical side and also from the interest in space and machine learning.
Thanks. Yeah, that's great that you get to apply it in an interdisciplinary way. So that's what we do here at FDL. Paul, do you want to go next?
Yeah, of course. So I'm a machine learning researcher, and this was just a fantastic opportunity, to do some really high impact interdisciplinary machine learning, some methods that I've not used before, and yeah, certainly in a challenging domain that I've not worked in before.
Thanks, Paul.
I'm going to say nothing new than what the other guys have said, but I think this product really does touch so many different fields. So, you know, we have the health element of it, but that health element, it ranges from, like, I guess what you think of more being like bioinformatics, looking at these really huge data set of genes, all the way up to, you know, thinking about cancer and thinking about disease in humans. But similarly, in the computer science stuff, we range from like really quite mathematical data science, which is coming up with these really important equations that govern causality, all the way to like very practical software engineering, which is how do we send data to different places when it's all federated and in different silos. And then we also have the considerations of, how do we integrate this into putting it on the ISS or putting it in a different aeronautical or astronautical environment. And there's so many things that come together in this product that I think are so interesting. And that's the real wonder of it, that genuinely multidisciplinary nature of it.
Yeah, I think that's so true. And you guys have so many opportunities with this challenge and the outcomes of this, so many different directions that it could be taken. So thank you. And I also wanted to hear from you guys. One of the most interesting things that you have learned from FDL, we're in week 5. You guys had a really intensive boot camp. You've gotten to interact with a lot of our different partners, from Google to Intel to NASA. So what is one thing that you guys learned or a fascinating fact?
If I can jump in here, great. I had a wonderful time learning more about radiation dynamics. So, the wonderful thing about having NASA as one of our partners is we got to sit down with, the world leading experts on space radiation. And it's fascinating to hear about, how we can try and simulate space radiation on Earth if we want to kind of, get experiments that get us good data. It's that, we use irradiated cobalt plates, I believe. It's just stuff you don't really think about, like how do you generate cosmic radiation on Earth? And it's fascinating, absolutely fascinating.
Yeah, that is really interesting to think about. That's awesome. Giuseppe?
Yeah, and also, so as Oren has just explained, the part of the biology is amazing, what's actually happening behind there. But I think from my point of view, I was amazed by the reality of this need of federated learning. So I always thought that there's this federating, it's usually used for privacy, but I never realized how actually in space there's really a lack of information you can transmit between, for example, space shuttle to Earth. And I find it amazing that with these tools we can kind of circumvent these issues. And I'd never realized how practical these methods are and how important they are in reality, and probably will be central in the future.
Yeah, that's a great point. Linus?
So I think one of the cool things about FDL is that we're also a little bit exposed to the topics of the other groups and the challenges that they are tackling. And one of the things that I learned there that I didn't really know so much about before is that space weather is actually something that's happening all the time. I was aware that sometimes there are events on the sun, like a solar storm, but I thought maybe that happens every couple of years. But you can actually go on the internet and look up a space weather forecast for tomorrow or for next week. So that's something like a fun fact that I learned during FDL.
That's awesome. Yeah, that's a great point, that we definitely interact across all of our different teams, and we learn so much from the other groups too. So thank you. And Paul.
Yes, I suppose I'll go slightly general. Just how challenging space is, and how it's like there are similarities to research that is conducted on Earth. Obviously, there's kind of the cancer link, being impacted with the radiation, but kind of the timescales, the radiation levels, like everything's just hyped up a level with space travel. So I think I'd never really considered how kind of challenging it is to do this.
Yeah, it's a really hard problem, but I'm glad that you guys are working on solving some of these. So great. And I also wanted to see, since it is week 5, you guys are preparing for a review. Are there any initial results that you can show the audience?
Well, I believe we can.
Great.
I'd say, these works are not final, so we can show you what we're investigating and what we think is potentially interesting. So what I can show you here are some potential genes that we pulled out from our combined human mass data. And as I said, we still don't know whether or not these are the best possible things they could be, because we have no ground truth for these data. We can't pull out a thing and say, ask someone, hey, does this cause cancer or not? But interestingly, of the genes we've pulled out from this sort of blended human mass data set, we've already identified 2 that have already been quite strongly associated with cancers. So these are TFF3 and GSTA1. And we know these are quite involved in cancer generating processes. And so if this, this model had 2,000 genes fed through it, and it pulled out of these in the top 10, and this does leave us to think that like we should investigate what other genes have been pulled out by this model as well. Particularly looking at things like TRIM10. And so if we, have this result already here, this is really good for us. We've also been looking at the similarity between mice and human data sets when we run analysis. This probably doesn't make this much sense. I'll try and explain what's going on here. So these numbers here are percentages, and so that one is 100%. And so these are different runs when we vary results with different humans in them. We want to see how consistent our results are. This tells us whether or not our method can work, even when we're looking at humans in different environments and in different settings. And the good news is, broadly speaking, our results are quite consistent across different, basically, groups of humans. And so it gives us more faith that the things we're pulling out in these sort of models are the things we expect to pull out all the time if we had human data and if we had mice data. So this is like these are the initial results we have that we can show off at this stage.
Awesome. Thank you for giving us a little bit of a teaser of what's to come and sharing some of the initial results that you guys have gotten. You still have a few more weeks left, so I'm really looking forward to seeing what you guys end up with. And I think we're coming up to our time here. I just wanted to see if you guys had any final closing thoughts to leave the audience with.
I suppose whilst there's no kind of graphs or results as such from the federated learning side, we have kind of a proof of concept pipeline that federates some of these causal methods, and we're kind of in the process of comparing them to the non-federated versions. So yeah, I can't show any fancy graphs, unfortunately.
That's okay, Paul. We'll look forward to looking at those and your results on the federated learning piece at the end, and we'll have a showcase and this will all be made public. So stay tuned on that one, but thanks for the quick update on federated learning. Anything else? Yeah.
On mine, I can talk about how, the coolest thing about this sort of stuff is that it makes like healthcare on Mars kind of like a more slightly less theoretical. Because with Federation, we have the potential to send models to people, light years away, and we can get the data back without having to send, all their data over. But similarly, if there's distributional shift between, the realities of living on Mars versus living on Earth or living on the Moon or living on the ISS, the models that we build try and deal with that distributional shift. And so this stuff really is space age, and that's wonderful as far as we're concerned.
Yeah, that's a great point. What you guys are doing really has a real world or off world practical application. Thanks. All right. Well, I wanted to say thank you to our astronaut health team. You guys have been absolutely wonderful, and it's been great to hear about your challenge. Looking forward to seeing the final results. Thank you to our audience for joining in with us today. And we will be back next week with a SETI Live with some of our other FDL teams. So thank you all very much, and looking forward to next time.
Thanks.